Hi, my name is Jonathan Rebhan. I am lead developer of swiftReg that has been developed in collaboration with Tim Mossman, Gaurav Sharma, and Sally Quadert at the University of Rochester. And I want to talk a little bit about how swiftReg might help you analyze and pre-process flow cytometry data to remove batch effects between experiments. So some key takeaways, we'll talk about some sources of batch effects and the idea of how registration can be used to help correct for some of those misalignments. We can look at some of the expected results. I'll talk about a little bit of the terminology that will be used throughout the other videos, and then we can summarize the best practices. So what are batch effects? Batch effects are non-treatment related factors that enter into experiments that are often the result of technical variation in collection procedures or in preparation procedures when experiments are run over multiple days. Some of these sources might arise from machine or experimental variation, and they typically have the highest contribution to the variability of results, and they often can't be randomized away. This is an experiment that was captured and sent to different laboratories, and it's comparing the results of centralized analysis versus analyses that were done in multiple laboratories. And we can see that in the centralized analysis there is approximately fourfold disagreement, but when the analyses were done across multiple laboratories there is approximately 50-fold disagreement in the results. And one of the contributing factors is likely the subjectivity involved in selecting gates when analyzing these data sets. And we have, for example, all of this variability is present across the experiments and files, and we want to find automated, reliable, and safe ways to reduce some of that variability so that the analyses of these data sets will be more consistent. So how do we correct these misalignments? Well, one of the considerations was borrowing an idea from imaging science, where satellite images needed to be arranged in such a way that the locations could be consistent and identifiable across many different sets of similar images. And when we extended this to more dimensions, we have a set of several bivariate plots. And the blue sample is our target, and the orange sample is our test sample. And you'll see that the test sample starts initially very misaligned. And over time, through many iterations, the mapping will register the misaligned clusters towards the target sample. So the results that you'd expect, in this case, this is a Cytof data set, and the upper panel is the raw data and the lower panel is the registered data. And of interest are these for the cell length, CD45RA, DNA1 and DNA2. You can see misalignment in the DNA channels and some misalignment in cell length and CD45RA. And after registration is applied, the four channels are better aligned, and we can apply one consistent analysis now across that entire experiment. So for our purposes, and as we get into later videos on the registration process, swiftReg requires a little bit of terminology. So when we refer to grouping a sample, multiple samples into a single source, we'll call that an aggregate or a consensus or a concatenate. And this is essentially just subsampling from multiple samples and aggregating them. We also will refer to internal standards or internal controls, and that would be an example of one sample that might be frozen down, and aliquots would be given to each experiment to be run in tandem with the experiment on a particular day. And then we have references. The reference sample is the intended idealized sample that we want other samples to be registered to. We have two different types of cluster movement. One is neighbor-dependent cluster registration. We call that NDCR inside the program. And that uses channel-specific movement that is designed to preserve relationships between clusters throughout the data set, and it only adjusts on a channel-by-channel basis. And I'll show a better example of this in the next slide. And then there's independent cluster registration, which allows clusters to move freely. It is not rank order preserving, so clusters can move past one another to find the best fits within individual channels. And here are two examples side by side. So in the top panel, it shows the same data that is raw, not registered. And what the neighbor-dependent cluster registration does is it estimates a best fit line through the population, and then it uses the best fit line to determine the size of the shift at the various points along that data to adjust for the differences between the target and the test sample. And for independent cluster registration, each individual point is allowed to move as much as it needs to match between the test sample and the target sample. And the two panels on the bottom represent the end product. So for the NDCR, you have the bulk of the population moving, but it leaves the order and the orientation between clusters intact, whereas in the independent cluster registration, it moves everything onto the line, indicating that there is a very tight mapping between the test sample and the reference. So for our registration objectives, we also consider individual registration or batch registration. Individual is taking one target or reference and then register the individual samples directly to that reference. For batch registration, we want to find a consensus representation of a batch. So this could be, for example, we could use an internal control that was run on the same day as that batch and use that to register to the reference sample. So for example, if we had one cluster, and this is showing 18 subjects, so each of these clusters is a different subject, but it all represents the same cluster. We might have 3 different batches, here indicated by the green, red, and blue. And if we broke this down according to the demographics of the sample metadata, we would find that some of these samples came from old or young subjects, indicated by the orange or purple. And we can register these batches according to the centroids of that batch. So for example, in the blue batch, it would have one centroid derived from all of these positions. And we move that centroid, we find its vector, the difference between that centroid and the reference, the red batch is our reference in this case. And then we move all of the blue clusters according to this vector that would position our blue centroid onto our red centroid. And then we can repeat the same process for the green batch. And once we've done that, we have all of the batches aligned, and then we can apply our metadata filter to find out where the old populations went and where the young populations went. So if we were gating on this data after registration, it would be much easier to organize the gates and apply them across all samples. Templates are another term that we use. The first refers to a reference cluster template, and that describes the positions, shapes, and proportions of the reference sample. And then we have batch shift templates, which describe the shifts that are required to align each individual sample according to its batch movement vectors to align that individual sample with the reference. What best practices do we recommend? Our recommendations address 4 different scenarios. In the first, if you want to align experiments run on multiple days and you have internal standards, then you'd want to preserve the variation within the batches using those internal standards. We recommend batch registration, and the method is NDCR followed by ICR. That means it's several rounds of neighbor-dependent cluster registration followed by a few rounds of independent cluster registration. If you want to reduce all of your MFI variation due to artifactual or biological differences, then we recommend just doing individual registration. And again, you can use the neighbor-dependent registration and then followed by independent cluster registration. If you want to remove channel-specific effects in the absence of internal standards, you would use aggregates of samples within a batch, and that becomes, in effect, your internal standard. You can use individual registration or batch registration, but in this case, we recommend NDCR only. And if you want to align your data when some batches contain subpopulations that are completely absent in other batches, then we, again, leave the distinction between individual or batch registration up to you, and again, recommend just using neighbor-dependent cluster registration.